think you signed it or they actually did
Pass those back and this one's for you. That's right.
I thought you were the professor. I'm like,
No, I didn't want to eat at the table, you know. Look, it's funny. I
Yeah.
you know, teach nursing students. That's right. When we were reviewing histology yesterday, I didn't know anything. No, this was Edgar. She's in your season. For level 4. No, this time they're not the best. No, this I hate to teach one, man. So, that like maybe one person paying attention at a time.
You got one person?
Yeah.
one person's paying attention, everyone else is just not paying attention at all. And they need this because they're nursing majors, but they don't listen.
Okay, well, let's get started. Again, they stuck us in not the greatest room ever for a class that is this full. So this is endocrinology. It's actually unusual for a class because it's pretty much split 50-50 between undergrad and graduate level. So this is our book, right? Vertebrate Endocrinology. It's a good book, especially those of you who are going on to medical type applications. This book will actually go even in more detail than we'll generally do. So it's a good book. I really recommend getting it. The 5th edition is fine. I think there might be a 6th edition. You can get used versions of this. I've had people find copies for as little as $5. So there's not really a reason not to have the book. And I will try to use figures and text from the book to make that a little bit easier. So I recognize a lot of you, but for those of you who don't know me, I'm Dr. Rogers. The easiest place to get a hold of me is here. This is my office phone. Unless it's during office hours, I'm probably not going to be there. Science Annex is my office. I'm guessing most of you know where that is. It's the parking deck looking building across from Kell Hall. I actually had a hard time. You can't get through Kell Hall anymore. So I had to try to figure out how to get over here. I'm getting the impression that it's going to be pretty warm in here, as a general rule. So there's that. We'll just have to deal with it. Maybe we get facilities to, it's going to be tough to get them to turn the AC on in the middle of January, these old buildings. Okay, so we meet 12:45 to 2:30. This is a 4-credit class. My office hours, the easiest time I'm in there usually from 3 to 5 on Monday and Wednesday. That's kind of an odd time, but I'll be in there those 2 hours. You can also, we can meet after class. If you want to meet after class, just tell me before you leave because I may or may not go back to my office. I'm here on Fridays as well, so if you need an appointment outside of either of those times, probably Friday is going to be the easiest time to make that happen. So this is, we said 4240, so we're kind of teaching it in the opposite sequence of uses. So I've got maybe 20% of my students in here from 4241, hormones and behavior. So conceivably from the numbers, you can indicate that this should have been the first course to take, and then hormones and behavior after that. But you know, with the way that the biology curriculum is, you just pretty much have to fit in your courses where you can get them. So for those of you who have taken hormones and behavior, there will be some overlap that you'll probably notice. But hopefully, certainly this is a course all on its own. So the reading is on the syllabus. as those of you who have had me before know, I do lecture a lot, right? A lot of the material is in the PowerPoints, but the PowerPoints are mostly slides, right? Pictures and images. So if you take great notes and you have the book as a reference, you should be fine. Again, I encourage you to get the book. It is a good book. Tests. So for an upper division course, this is done a little bit, at least as far as I know, a little bit differently. So there's 3 exams through the course of the semester, and then there is an optional final as well. The final is comprehensive, obviously, but if you've taken all 3 of the other exams and you're happy with your score, you don't need to take the final, right? If you choose to take the final, right, you can improve your score, but you are by no means obligated to do so. And if you're doing well in the course, you probably have other things to study for. But it's there if you need it. Exams, so they're, let's see, so the course is a lot of how the endocrine system works, how it functions, right? So there is a lot of content, right? If you're unfamiliar with it, you will kind of have to learn the vocabulary of endocrinology. There will be some neuroanatomy, right? But generally, this is not going to be an essay-based test, right? We're looking at multiple choice, fill in the blanks, matching, short answer types of tests, right? You're not getting page-long essays. There might be 1 essay on an exam, but that would probably, traditionally, that's about as much as you're going to get, right? This is, do you know how the endocrine system works? Do you know the parts? Do you know the receptors? Do you know the effects, right? This is very focused in that particular way. So for the grading, it's actually pretty straightforward, right? We have 3 exams, right? You can potentially take 4, but only 3. Your best 3 will count. There will be 2 out-of-class assignments. Those will be reading 2 different primary research papers, and you will, with a partner, present a paper of your choosing on one of these topics. The time at which you do that will be at the 11th of April. And we can, right, you'll have to get your paper approved, and we will go through that, and I'll give you a rubric for the presentations and all of that good stuff. This is a 4,000 level class. Your attendance is expected, right? Those of you, and traditionally, right, this course works best when it's participatory, right? And so if you participate, I get to know you, right? And if I get to know you, when it comes time, I can add participation points to your score, right? That's the way that works. If I don't know you, I know you haven't been participating, right? There's only 36 of you, which is fairly decent size. I will get to know most of you if you talk a lot, right? Which is good. So if you start missing a bunch of classes, right, we're gonna have an issue, right? This is a 4,000, 6,000 level class, right? You should, at this point, know what you need to do to be successful in a course. If you don't know, I'm here to help you. If you miss an exam, you can take the final as a makeup. I think that is pretty much it. So, nuts and bolts, any questions? Okay, great. All right, so I guess we'll just go ahead. We're not going to take the whole time today, but I want to go over a few kind of really key concepts for understanding endocrinology in general. And so most of this is going to be review, I would suppose, but I really want you to start thinking about this from the perspective of the endocrine system, even if it is review. And this is the idea, we've got chemical signaling and bioregulation, right? So chemical signaling, right? So this is information, right? And we're going to talk about the different routes of information, but a signal is informative, right? Now, ideally, when a signal is sent, right, there is essentially an ostensible target. But what we'll learn with much of the endocrine system, because those signals are in the bloodstream, right, all tissues could potentially respond to that, right? And so that informational signal might be useful to coordinate, right, different responses physiologically. The example I really like to use is a hormone, right, that you've probably all heard of, melatonin, right? So what is melatonin, for those of you who know? I hear a lot of people saying sleep, and I heard some people saying circadian rhythm, right? So those are really different things, aren't they? Right? And I think that's great that we both figured. So a circadian rhythm, right, is basically your 24-hour, right, physiological processes, right? And sleep is a specific thing. It's a specific state, right? So, right, melatonin, when is melatonin released? At night, conceivably, right? So how does the body know that it's nighttime?
And numbers, photons.
Right, photons, right? So an absence, actually, of any kind of retinal signal is going to, right? So that's what our system uses, right? If you have a retinal signal, right? And we'll talk about the pineal gland a lot, but basically when you have light, right, that suppresses the pineal gland, right? When it is dark, that inhibitory signal isn't there anymore, right? So melatonin, which is what's known as an indolamine, it's a serotonin-derived hormone, right? It begins to be secreted into the bloodstream. Now, what does that tell us that we should be doing? Sleeping, because we are diurnal animals, right? But the signal is informational, right? Do nocturnal animals have melatonin? Ooh, interesting. Yes, they do have melatonin, right? They have a pineal gland, and it has the same organizational structure that ours does. So when it is light, right, melatonin is suppressed. When it is dark, melatonin is released. Let's give me an example of a nocturnal animal. An owl, or say rabbit? Bat, okay. Rat, also, rhymes with bat, right? Okay, so what does melatonin tell those animals to do?
Wake up.
To wake up, right? And again, these are, right, so tissues respond to the hormone signal that it's dark out, right? That is an informational signal that synchronizes your circadian rhythm, right? The signal itself doesn't tell you to wake up or to go to sleep, right? But the response of the tissues in charge of wakefulness, right, and rest respond to the signal in very different ways, right? So you have a chemical message, right, that is informational that can then be interpreted by the specific target tissue, right? So we talk about circadian rhythm. So when do you get your circadian rhythm thrown off? When is your, right? So normally you wake up, you go to sleep on a regular rhythm. When does that get thrown off? Jet lag, travel, right? When you go to a place that is different, right? Melatonin itself doesn't seem to be very efficacious in getting you to sleep, but it's really good at synchronizing your circadian rhythm, and a byproduct of that is that you then are better able to sleep in your new environment, right? Because the signal is informational, right? If you put yourself in a dark room and take melatonin, right, you're essentially resetting your circadian rhythm. Okay, so again, so this is a chemical signal, it's informational. So as we'll kind of do throughout this course, what happens when that situation or that normal pathway is broken, right? When could we imagine that a person has a disorder of melatonin? What is that person also going to have? They're going to have circadian rhythms. Why are they going to? They're going to have circadian rhythm problems. Why are they going to have that? What are they missing?
They don't know what to do.
They're blind.
Oh.
Right? I mean, you can have pineal disorders, sure, but the most common disorder of melatonin, just because we're talking about it, right, is blindness. Right? Now, you might say, what does that mean? Well, you know, even a blind person is going to want to wake up and go to sleep on the same pattern that we all are doing, right? But they have melatonin being released the entire time, right? Because there's nothing to inhibit melatonin. So in order to sort of solve these problems, they have to synchronize in very different ways, right? So if you blind an animal, they will have what's known as free running sleep, right? You get tired at the same time every day, more or less, right? Have you noticed this? Now, if I were to deprive your body of signals, it wouldn't take too long, or if you were in a cave or something, right? For you to stop sleeping at the same time each day, and your day-wake cycle would be what's known as free running, and we'll look at experiments that they did in rats to show that. Okay, so that's chemical signaling, and again, this is just an overview. We're going to talk a lot about chemical signaling and a lot about what the different chemical messages are. And the other piece is bioregulation. And this, I like to think of it, the sort of fundamental principle of endocrinology is homeostasis, right? The whole key to a kind of free and independent life as an organism is the ability to maintain your internal environment. And the larger you are as an animal, right, and the more complicated you are as an animal, the more you have to synchronize across different systems. And so this process of maintaining an internal environment, which occurs at the cellular, the tissue, the organ system, and the organismal level, is all about maintaining that internal environment. So we're talking about chemical communication a little bit. Again, if you haven't seen something like this by this point, your teachers have failed you. It isn't to say that you have, but seriously, you should have run across this at some point. Right? We basically got, you could also add in sort of terracrine, but this is the type of signal, right, kind of overlaid on its route of administration. So we've got three, right? We've got endocrine glands, we've got neurohormones, and neurotransmitters, okay? So neurotransmitters, we'll talk, here and there about neurotransmitters because, right, obviously the endocrine system is controlled by the nervous system in large part, and that involves neurotransmitters. So neurotransmitters are just chemical messages, right, that are secreted across a synapse, generally targeting another neuron, although not always, right? A neurohormone is a chemical message that is produced by a neuron that is released into the blood, right? So these can be things we normally think of as neurotransmitters like dopamine and serotonin, but they can also be things like right? Oxytocin, vasopressin, right? These are things that are produced by neurons. These are, you'll see this, right, vocabulary, neurosecretory cell, right? This is a neuron that produces things to be released into the bloodstream, right? Not all neurons, right? Not all nerve cells are neurosecretory cells, but all neurosecretory cells are neurons, right? And then you have endocrine glands. Endocrine glands release their products into the bloodstream that is then carried to all of the other body's tissues, right? They'll have access to it. And again, these are not derived from neural tissue, right? These are, they can be different types of tissue. A lot of times they're epithelial tissue, but they release their products, right? Usually through vesicular release into the bloodstream. Okay, so when you think of chemical bioregulation, right, the endocrine system, the immune system, and the nervous system communicate with each other, right? And so some of the things we're going to spend a lot of time over the course of the semester is talking about the crosstalk between these systems, right? For example, right, the nervous system activates the endocrine system, right? And you can even think of things like your digestive tract, right? If you sever, right, if you sever the nerves that go to the pancreas, right, and you eat a chocolate sundae, will the pancreas produce insulin? Any A&P TAs in here? It will, actually. It will. The pancreas itself has what's known as a glucostat, right? And so your pancreas has specific glucose transporters, which it is constantly taking up glucose out of the blood, right? And we'll talk in great detail how the glucostat works, right? And so if you did nothing else, right, your pancreas will respond to an increase in sugar without any input from the brain. But, right, if you look at a chocolate sundae, like if you put a donut out here, I haven't had lunch yet, so if you put a nice sandwich or a donut out here, my brain sees that, and it's going to stimulate, right, the release of insulin before I start to consume that food, right? If I sever the nerves to the pancreas, right, the endocrine system can still work, but it's not going to be as effective, right? So there's crosstalk between these systems, right? It's much the same way, right, as the heart, right? If I sever the nerves to the heart, right, will it keep beating? Yeah. Yes, it will. Right? But do you have nervous system interaction in there? Yes. You have some, yeah. Yeah. And you have some endocrine interaction there too, right? So if I load your system with epinephrine, right? And let's also think, so where does epinephrine come from? Yeah, the adrenal medulla, which is part of your sympathetic nervous system, right? So epinephrine is released. It has receptors on your heart, right? That will change your heart rate. And so, again, I mean, these are kind of just simple ones that we kind of know, but when we talk about these systems and these regulatory networks, it's important to keep in mind that they talk to each other, right? And so, kind of keep on that theme of chemical communication, right? We've got a bunch of different kinds of cells. We've got our neurosecretory neuron. We've got what we're calling ordinary neurons. We've got endocrine cells. We have cells that are autocrine. These are often immune cells. And then we have this sort of last part, which we haven't talked yet about, but about metabolism and excretion. So ordinary neurons, right, they release neurotransmitters and neuromodulators. So you'll see this as kind of a gray area. What's the difference between a neurotransmitter and a neuromodulator?
Neurotransmitters have to be made in the vesicle, right? Or stored in vesicle, or like, no, built. Shit, we just had neurobio last semester. All right, so neurotransmitters, they have to be constructed like in the bouton or whatever, you know, the end of the axon, and they have to be stored in vesicles, and then they have to be secreted into the synapse and onto their respective receptor.
Okay, and so what's a neuromodulator?
It doesn't necessarily have to be made in the body, like alcohol is a neuromodulator.
That's not how we mean it here, right? So that, and again, it's really, this is more of a vocabulary type issue. So when I say neuromodulator, I mean something specific, right? So there are lots of compounds which modulate neurons, but in the context of, right, the release of a neuromodulator, I want to be specific, because, as specific as I can. Because let's say you take something, right, like, we'll take one like serotonin, someone that you guys are probably familiar with, right? So serotonin can be released as a neurotransmitter, right? It's packaged, right? Both, and the point I wanted to get to is neurotransmitters and neuromodulators, and in fact neurohormones are all vesicularly released, right? The specific type of chemical that you're making is going to affect how they're made and packaged. But so a neuromodulator generally refers to, right, neurotransmitters that have actions outside of the synapse itself. And I'll give you an example of this, right? So everybody understands what a synapse is, right? Okay. If you don't, and you don't feel like raising your hand, that's okay, you can ask me later. But a synapse is basically a small space, right, where the receptors are right up next to, right, the presynaptic neuron. Okay, so I guess I'll illustrate this by way of example. Everyone said serotonin, right? Serotonin can be released synaptically oftentimes. But serotonin also works as what's called a neuromodulator, right? So has anybody ever heard of an SSRI? Okay, what does an SSRI, so give me an example of an SSRI, and what does it do? How does it work? Zoloft, okay.
Selective serotonin reuptake inhibitor.
Keeps
Oh.
it from uptake.
Okay, right, so it's right there in the name, it blocks. So when we talk about metabolism and excretion, and so I can tell some of you have had neurobiology, basically when a vesicle is released, right, it basically has a clock on it. We measure that in terms of its half-life, right? Different kinds of chemical messages are broken down at different times. So like long, big proteins, right, like growth hormone, can last for hours, whereas something like serotonin might only last moments or minutes or less in some cases. Okay, and so one of the ways that many neurotransmitters and neuromodulators are regulated is once they're released into the synapse, they are taken back up by what are known as these reuptake transporters. So serotonin's released, and basically as soon as it's released, the transporters start taking it back up again, right? That's what contributes to its half-life. And then it can either be broken down or recycled into another vesicle. Okay, so an SSRI blocks that. Now, how long, on average, does anybody know, how long does it take for an SSRI to have its therapeutic effect?
About 2 weeks.
About 2 weeks. How long does it take to block that transporter after you take serotonin? Or if you take the SSRI, how long does it take before it blocks the transporter?
Hours?
Okay, let's say it's an hour. I don't know the exact number, but I think it's shorter than that, but let's say it's about an hour, right? We can compare it and contrast it with another, right? The dopamine blocker, right? That works the exact same way, right? Has anyone heard of cocaine? Cocaine blocks the same transporter, but for dopamine. How long does that take to block the transporter? It's let's just say super fast, I've heard. I've heard it's pretty quick. So, these are pharmacological agents. The effect, let's say worst case scenario, you've blocked, right, your transporter in an hour. But it takes 2 weeks of constantly taking that transporter blocker for you to have an effect. Why?
At first, when it starts to be not taking, even but people later watch. So you have to be systematic.
Right. So the short answer is nobody really knows why, but there's lots of speculation that so in the brain we have what's known as neuromodulatory tone. So there is background dopamine and background serotonin that are acting at serotonin and dopamine receptors, which are not in the synapse, which are distributed throughout the neural network itself. And so a neuromodulator is essentially a neurotransmitter that has a more paracrine effect, right? It affects a larger area, right? So the therapeutic effects of an SSRI are not synaptic, right? These are, and we can know that because you can block the receptors and you have no effect on the phenotype. It takes time for the phenotype to emerge, right? So it takes time for you to accumulate this new neuromodulatory tone. So I said it was a sharp definition, but it's really more of a fuzzy one, right? Things like glutamate and GABA are not neuromodulators. Things like serotonin, right? Melatonin, dopamine, right? Norepinephrine, these can be neuromodulators, right? So neurohormones we've already talked about. Hormones I hope we've talked about. Autocrine, this just means kind of self-regulating, right? So if you, right, take a drug of abuse, does that dose always do it for you? No. And generally speaking, when an addict stops taking a drug, what happens to them? There's withdrawal. Why? Well, let's take the endocrine kind of, right, homeostatic approach, right? So let's say you are essentially, right, trying to regulate the amount of dopamine in the system, right? So we have a given amount of dopamine. So if I give you cocaine, what happens to the amount of dopamine available to the synapse? It goes up. If I start taking a lot of it, what's going to happen to my endogenous dopamine? I'm going to stop making it. I'm going to make a lot less, right? Because I'm trying to recover to my set point. We'll talk about set points in a minute. I'm trying to recover to my set point, so I'm going to make less. I'm going to make less and less and less. The more you give the system, the more the system is going to respond by making less. So then what happens when I take it away? Oh my God, nothing will make you feel positive because your system will not make enough dopamine to meet all of the new receptor thresholds, right? So this is part of this kind of homeostatic process. So how does, to get back to our paracrine question, how does that dopamine neuron know how much dopamine it should be making? Well, one way it knows is by sampling how much dopamine is around it, right? By these autoreceptors, right? So if I'm a dopaminergic neuron, I have dopamine receptors on my synaptic terminal. Why? Because that way I can make sure that I don't flood the system, or if my receptors aren't being bound enough, I can make more, right? And so if we kind of spin that forward over time, if I have no stimulation from dopamine, now my neuron is going to start making more, right, to get back to that set point. The last thing I want to talk about here on this slide is metabolism. We've kind of already started talking about this. So every chemical messenger has a half-life, right? And the reason for this is pretty straightforward. If you want to send a signal, if I want to send you an informational signal and I just set a tone and I just kept that tone inevitably, forever, well, how much information could I give you?
Only the initial.
Only the initial, right? Only the initial signal has value if the signal isn't, right, doesn't have some cessation, right? Any informational signal has to stop, or I can never give any more signal information along that line, right? So if you think about the way that neurons work, right, they work on the order of milliseconds to fire, sometimes, right, even shorter time periods than that. So if I'm a neuron and I am sending a chemical message, right, at 60 hertz, which is not unheard of for the nervous system, that means I'm releasing neurotransmitters 60 times a minute. Now, that means, right, and there are only certain, transmitters that this will work for, but that means that signal has to come and get cleared really, really, really fast, right? Reuptake is one of those signals, right? Enzymes, right, like acetylcholinesterase, right, in the neuromuscular junction. These are metabolizing the signal, right? The signal has to end, right? Everything has to have a half-life, and ultimately it's metabolized. Generally, that means an enzyme breaks it into a different chemical structure, which no longer activates the receptor. And we'll see in some cases these metabolites themselves may have different or off-target actions themselves, right? But generally, they are metabolized, they are converted into something which no longer has the primary effect, and then ultimately they're excreted, right? Usually via kidney. Okay. These are actually all figures from your book. Okay. So this is kind of a schematic representation of that, right? You have the endocrine cell, right? It releases its hormone. It's often carried, right, via chaperone proteins, right, through the blood to the target tissues where it can have its effect, right? It's metabolized by the liver, right? Ultimately excreted by the kidney. You have this auto-regulation situation here. And so what you can see is, and this is the idea that I want you to get to, right? When we talk about a hormone, let's say, right, vasopressin being released, right? Vasopressin has a half-life, right? If I want to continue to tell the kidneys to retain water, and that's what vasopressin does in the periphery, I have to keep releasing vasopressin, right? Now that's important, because once I reach the set point, I don't want to keep, right? And I don't want there to be a huge lag time when my kidneys are absorbing a bunch of water, right? Where I don't want them to be absorbing that water anymore. So in the absence of anything else, right, these hormones have to be broken down. Now, things have very different half-lives, right? So growth hormone, you don't necessarily want growth hormone to have a half-life in 2 to 3 minutes. Like, what sense would that make, right? But something like AVP, right, vasopressin, right, oxytocin, you want these things to have very short half-lives. Okay. So, last thing we're going to kind of talk about, and I'm not going to belabor it too much, is to talk about homeostasis. And primarily, endocrine regulation of body systems falls under, right, negative feedback, right? And we'll just go through negative feedback because there's a very important difference between the ways that people understand negative feedback, especially as it relates to the nervous system. Any discussion of homeostasis would not be complete without the founders of the idea, Claude Bernard, a very famous, right, biologist, and the first one, right, to talk about homeostasis, and then ultimately Walter Cannon, who, amongst other things, right, discovered shock and epinephrine and the relationship between, right, all of these factors and the maintenance of an internal environment. But I want to get to this, right? So negative feedback. Everybody knows what negative feedback is, right? Okay, somebody give me a short and sweet definition of what negative feedback is. Yes.
So the final product of any pathway could theoretically inhibit the initial starting of that pathway.
Or any other stuff.
Right, okay, that's any other step, yeah, okay. So that's a functional definition of how homeostasis does what homeostasis does. So what is homeostasis, what is negative feedback trying to do?
Not.
Maintain homeostasis, so there's something, right? All the different systems, right? So there is what we would call simply a set point. Yes?
Maintaining homeostasis by inhibiting the formation of a product.
Yeah, so homeostasis can take a lot of different, right, forms, and that's one of them, right? You can inhibit the formation of a product, but I want to give us some sort of basics of what's going on, and so then we can, because all of these things that you've mentioned are correct under certain circumstances, but homeostasis is quite variable in the way that it works, right? So there is, in negative feedback, and really even in positive feedback, there is a set point, right? This is a value, generally speaking, of some characteristic, right? That could be blood oxygen, blood CO2, blood sugar, right? pH, osmolarity, right? Temperature, right? So the really easy one is like 98.6, right? So, for example, with glucose, it's like 5 millimolar glucose is what your body wants. That's a set point, right? Now, in order to make this work, right, you have to have some kind of sensor, right? So the goal of negative feedback is to maintain the set point, right? So homeostasis is all about maintaining set points. Now, are those set points fixed forever? No. So generally they're not, right? So your body, and we'll do a whole thing on feeding and hunger and metabolism, right? Your body is going to fight really hard to maintain where it is right now. It's going to be hard to gain significant weight over where you are right now. A little easier, I find, than losing it, but still, if I wanted to gain like 30 pounds, that would take a lot of effort. My body does not want to gain 30 pounds, nor does it want to lose 30 pounds. But you can establish a new set point at either 30 pounds heavier or 30 pounds lighter, and your body will then fight to maintain that set point. Right? So the set point is not always fixed for everything, right? 98.6, that's pretty much it, right? You're not going to establish a set point outside of that, right? Blood glucose is another one. But, so, a sensor. So you have to have somewhere or some means of telling you where you are, right? So let's say you're at 10 millimole glucose. You need to be able to know that. Where's the sensor, right? Turns out for glucose, the sensor is actually in the pancreas, right? For temperature, the sensor is in the hypothalamus. And so you have to compare these two things. When they're the same, nothing happens. When they are not the same, right? So this is all sort of stored inside some, we'll just call this the controller. And there's like a fancier diagram here, right? Inputs and all this, but, right? The sensor and the set point have to be stored somewhere where they can be compared. Now, if your value gets too high, you do something. Right? You cause some effect if it's, so high, and then, you have a separate effect if it's too low, right? You have some effect that's generated. Okay, so when someone says, what's negative feedback, it's basically maintaining a set point. That's really all it is. Now, what that set point is actually depends on what you want to measure. So, here we go.
Okay.
So I want to give you two different versions of negative feedback. Let's look for the first one at fluid balance. So, fluid balance is essentially how much water you have. Right? And so, if the amount of water drops too low in your body, right, you want to correct that problem, right? So, the variable being measured is something called osmolarity. There's also baroreceptors. We'll get into this, but we'll just start with osmolarity and we'll just go from there. Okay? So, osmolarity is the measure. You have osmoreceptors in your hypothalamus. They are paying attention to the amount or to the osmolarity of your blood. If the osmolarity of your blood gets too high, what does that mean? Not enough fluid. All right? So, you're going to respond by releasing something called antidiuretic hormone, also known as arginine vasopressin. Right? This targets the kidney, right, the collecting duct, and distal tubule. And it basically tells the kidney to absorb more water. Right? So the effector is this antidiuretic hormone, or AVP. It tells the kidney to absorb more water. Now, when does this process stop?
When the osmolarity reaches the set point again.
Right. So what's being regulated? Is the amount of ADH being regulated? No, the osmolarity is being regulated, right? And there is the flip side, right? So if you drink too much water, if your osmolarity drops too low, what happens? You urinate a lot, right? So it works both ways, but the important point here is the variable, the set point, is for osmolarity. And a difference in osmolarity is going to trigger the hormone. The amount of hormone itself doesn't matter. Nobody is paying attention to how much ADH is there. Okay? Let's take another one. Let's take testosterone. Right? So, testosterone, you have this classic hypothalamus pituitary gland structure, right? So the hypothalamus is going to produce something called GnRH, right? That's going to cause the pituitary to secrete something called LH, which is going to cause the gonad to produce testosterone. Now, testosterone, and this is what you guys were talking about, actually feeds back both onto the pituitary and to the hypothalamus, which means that there are androgen receptors in the hypothalamus, right, in the arcuate and POA, and in, right, the gonadotropes of the pituitary. So this is a negative feedback control, right? But it is different. What is there a set point for? Testosterone. Right? So here you are regulating testosterone itself, and in the other example, you were regulating osmolarity. Right? So when does GnRH secretion stop? When testosterone is sufficiently high. And so we can see both of those are examples of maintaining a set point, but don't be confused that the set point is always the hormone. Right? In some cases it's the hormone, and in some cases it's not the hormone. So what happens to this system if you add extra testosterone to it? Okay, so you're gonna stop making GnRH, right? You're going to stop making LH, right? Because you're adding testosterone exogenously, right? What is that going to cause physiologically?
Oh, your testes are gonna shrink.
Exactly, right? Why are your testes going to shrink? Because they're not being stimulated by LH to produce testosterone.
Do some people add exogenous LH?
Yes. They don't usually add it as LH, they add it as something that's more readily available, right? Have anyone heard of HCG? So what is HCG?
Pregnancy hormone.
It is the pregnancy hormone. Right? What does HCG stand for?
Human
chorionic gonadotropin, right? So LH and its sort of partner in crime, FSH, are what are known as gonadotropins, right? So, and we'll talk a lot about pregnancy, right? But the job of HCG, as it's, okay, so HCG is produced by what? The placenta, right, or as the placenta is forming, right, the placenta itself becomes a temporary endocrine gland, right? Now, its job, one of its very first jobs, is to produce HCG to maintain mom's endocrine system. And so we'll talk about that, right? HCG binds to the LH receptor. And so, yes, people do, right? But generally they don't take LH directly, but they often take HCG, although the effect would be the same if you took either one. So what would happen if I did give you I didn't give you this, if I just gave you LH?
Produce more testosterone.
You would produce more testosterone. Exactly. Now, again, we're still going to run into the same feedback issue, right? You're going to stop producing GnRH, right? And you're going to stop producing LH on your own, but you're certainly not going to stop producing testosterone as long as you continue to give exogenous LH. Okay, so that's basically what I've shown here, right? You can kind of go through this. These are, right? So LH is what's known as a tropic hormone. We'll kind of go through all this. I kind of want to give you kind of an overview. This is a tropic hormone. This tells another gland what to do. And so here, right, the hormone is actually what's being affected, right? That's what this chain is. This side, A, is much more like the osmolarity. So both are actually examples of negative feedback. One, right, and in both cases you're regulating the set point, and the set point, however, is measuring something different, right? Okay, so if we look at this, right, this is basically the subdivisions of your endocrine system. We're going to kind of divide it, right, into the sort of neuroendocrine system, right? And I'll post these slides for you. And again, they're in your book, too, which you don't probably have yet, but it's worth getting as a kid. So the neuroendocrine, right, almost all of it involves the hypothalamus, the pituitary, usually some tropic hormones like LH, like growth hormone, like TSH, right? Those target endocrine glands that release hormones that have targets, right? So, like, what is the target tissue for testosterone?
Related, so.
No, they make it.
Oh, it's true. The target tissue.
Oh, okay.
Muscles.
Yeah, that's the target tissue for LH.
Yeah.
Muscles.
Muscles are one.
Bones.
Can be bone, brain, testosterone's everywhere, right? Testosterone. Where is the target tissue for AVP? It's in the kidney, right? Most of your other tissues don't respond, right? When AVP is released in the brain, right, as a neurohormone, it has effects in the brain, but generally speaking, the target tissue is going to be, right, the organ itself, right? So nonapeptides, these are AVP and oxytocin, right? The tropic hormones we'll talk about. And then you have some autonomous endocrine glands, right? The example, the best kind of example are in your digestive system and, right, your metabolic hormones, right? Like your pancreas, as we said before, it does not need the brain's help, but it works more efficiently connected to the brain. Okay, so we're breaking this down a little bit more, and I think I'm actually going to end on this slide today. I don't want to overwhelm you guys on the first day. But, right, so you have, right, the three divisions of the pituitary, right? You basically have all of the hormones that are produced from the neuroendocrine system, right? AVP, oxytocin, right? Melanocortin, growth hormone, prolactin, right? TSH, LH, FSH, right? Adrenocorticotropic hormone, and growth hormone, right? So we're going to spend the next couple of weeks unpacking all of this, especially here and at the level of the hypothalamus. So there will be a lot of, right, receptors, which is what we'll start with, like chemical messengers, manufacturing of peptide hormones, steroid hormones, all of that. And then we will move into the anatomy of the brain and what regions of the hypothalamus control the secretion of what hormones and so on. Okay, any questions before we go? Okay, so try to get your book, like I said, digital copy, hard copy, right? It's worth getting, I think, as a reference, especially those of you who will go on and either teach this or potentially practice this.